Okay. Could you see the presentation?
Yeah, it is visible.
Yes, sir.
One screen
Visible.
is visible.
It's okay? Okay, let's go. Well, hello, everyone. You are welcome to our first Scientific Committee on Cellular Therapies webinar today. I am going to offer you a brief introduction to the main speaker, Dr. Asawari, topic today. And the topic is exosomes. Exosomes are nanosite biovesicles released into surrounding body fluids upon fusion of multivesicular bodies and the plasma membrane. They were shown to carry cell-specific cargoes of proteins, lipids, and genetic materials, and can be selectively taken up by neighboring or distant cells far from their release, reprogramming the recipient cells upon their bioactive compounds. Therefore, the regulated formation of exosome, a specific makeup of their cargo, cell-targeting specificity are of immense biological interest, considering extremely high potential of exosomes as non-invasive diagnosis biomarkers as well as therapeutic nanocarriers. Scientific teams are focusing on the potential use of exosomes as valuable diagnostics and prognostic biomarkers. Here you can see hallmarks of exosomes. For example, regulation of gene transcription and translation, survival and proliferation, reproduction and development, apoptosis, cellular differentiation and neoplasia, cellular migration and metastatic disease, and more. In the past few years, an exponential increase in studies focused on the biological characteristics of exosomes. The exosomal secretion from various types of cells, existence in almost all kinds of body fluids, and function as mediators of cell-cell communication, which make exosomes play a crucial role in both physiological and pathological processes. Moreover, the capacity of exosomes to envelop a wide range of content, including lipids, RNAs, and proteins, to signal a specific recipient cells or tissue, makes them a promising diagnostic biomarker and therapeutic tool of treatment of cancer and other pathologies. Mesenchymal stem exosomes have been tested in various disease models, demonstrating a similar or even superior functional capacity by themselves, reducing myocardial infarction size, preventing adverse remodeling after myocardial ischemia or reperfusion injury, providing therapeutic effects in cutaneous wound healing, acute kidney injury, hepatic injury, or neonatal lung injury, promoting survival of retinal ganglion cells in optical nerve crash, or ameliorating retinal laser injury and orchestrating neurological protection by the transfer of MRIs. Recent progress of exosomal separation analysis and detection. You can see in the diagram the recognition and detection of exosomes, and the focus is on nucleic acids, surface proteins, lipid localization, surface modification, for example, by radioactive material or nucleic acid recognition, other styles like microfluidic chips, or the overall recognition, for example, by flow cytometry or imaging analysis. In this picture, you can see a short list of exosomal bioactive molecules in cancer. For example, the microRNAs, in this case, these examples show you for example, the 122, promotes metastasis. Other ones promote oncogenic activity in gastric cancer, or confers doxorubicin resistance and tumorigenesis. In the case of these examples of long non-coding RNAs, for example, promotes cisplatin resistance, promotes cell proliferation, clonogenic growth, and tumor growth. And in the case of this example of proteins like PD-L1 inhibits immune response, promotes tumor growth, or the TNF-alpha induce immune cells death. Due to their role in cancer progression and biological features, exosome possess promising potential for cancer therapy. Options: applying naturally to suppress cancer cells, inhibiting cancer cells derived from exosomal activity, and using exosomes as gene or drug carriers. However, there are considerable challenges to be solved. First, the difference among exosomes from different sources is still not clear. Second, the exosome number to get a therapeutic effect may be significantly different among different cancers. And third, the scalability and the heterogeneity of tumor may influence therapeutic outcome. Clinical trials of exosome-based cancer therapy are urgently needed to determine the efficacy of the application. While there are still challenges ahead, it's clear that exosomes offer novel and important applications for the treatment of cancer. And the question is, are exosomes the future of stem cell therapy? Well, our main speaker probably in the next minutes could show us the response. The conclusions. Exosomes have extracellular vesicles that produce by mesenchymal stem cells and are involved in cell communication. Other cells react to the messaging signals from the exosomes and will change their behavior accordingly. Exosomes are so small and nimble and can remain hidden in the bloodstream and carry multiple doses of proteins across barriers that cells are not able to cross. My point of view, exosome research is now in its infancy, in-depth understanding of cellular components and mechanisms involved in exosome formation and specific cell targeting will bring light on their physiological activities. Well, thank you. Please, Dr. Aikaf, could you present our main speaker today? Thanks.
Thank you, Claudio, for the wonderful introduction to the subject.
You're welcome, Nasa.
Hi, good morning, good afternoon, good evening. My name is Dr. Asawari Bapat, and I will be speaking on exosomal therapy, a new frontier in regenerative medicine. I am a global regenerative consultant, a clinical pathologist, and a hospital and healthcare management specialist based here in Dubai and in the US. This is my brief bio. I have 20 plus years of leadership experience in managing and operating facilities globally, diagnostic labs and healthcare facilities, cord blood banks, adult stem cells, and tissue banks, facilities for regenerative medicine, gene and cellular therapies, which are currently called as biotherapies. I'm a globally validated subject matter expert for clinical trials and therapies, orthobiologics and devices, specialized in quality management systems, regulatory compliance, regulatory affairs, gap audits. I am also a faculty for conducting training and medical education, consultant for cell and gene therapies, biotechs, pharmaceuticals, regenerative medicine facilities, and companies globally. I represent on many of the committees and coordinating bodies as global subject matter expert for cell and gene therapies, such as Standards Coordinating Body, which is the Standards Developing Committee for companies such as ISO, for various governments, for various accreditation bodies, for European Union directives, for cell and gene therapies. I'm the elected member of CTSCC, which is the Cell Therapy Subsection Coordinating Committee, at the AABB, which has been recently branded as Association for Advancement of Blood and Biotherapies, co-chair of Asia Pacific Group International Variance Committee, the CT or Cell Therapy Standards Committee, where we are currently drafting the new cell therapy standards. I'm the international ambassador of ABB since 2015. I'm also the chairperson of AATM, UAE chapter. I'm the chair, speaker, moderator, a panelist at various international conferences and meetings. I've been published and have written chapters of books and books. These are my disclosures. Sorry, I have no financial disclosure and no conflict of interest at the time of this presentation. I do, however, have financial and professional relationships with the following organizations, such as ABB. I'm an advisor to Parents Guide to Cord Blood, IFATS, which is the International Federation of Adipose Therapeutics and Science, Regenerative Outcomes. I'm a faculty for ABRM, MENA, and other similar bodies, and a consultant to global cell and gene therapy facilities. This is the overview that we would be really touching on and looking at exosomes in a nutshell, and what are the new frontiers that we would be dealing with and we would be focusing on in the future. So to learn something basic about exosomes, I think we all learned in our schools what exosomes were and extracellular vesicles were. And it used to be thought that these are something like a waste product which is being removed out of the body. But it's only recently in 2007, a group of sort of scientists or researchers in Holland found out that exosomes also contain proteins such as RNA, DNA, and have other substances which are the parts of the cells that secrete them. And these characteristics and behavior of the exosomes closely relate to the parent cell of origin because of that. And there's an intercellular communication which is involved, and this affects cell function and cell behavior. Almost all cells, tissues, and body fluids secrete them, and it is involved in the pathogenesis of various disorders, including cancer, neurodegeneration, and inflammatory diseases. It is being widely researched currently, and there are so many articles which are being published, and around 253 clinical trials directly or indirectly, which are looking at the therapeutic applications. Sorry. When the pandemic started, we actually, I think around 2019, we were doing consulting for some of the exosome companies. And I will be mentioning some of them, but not. I just wanted to present all this information as a non-biased or a neutral person to you. So exosomal therapy, a new frontier in regenerative medicine. And what we found was, on account of their size and property, with the evident role in numerous pathobiological processes, the researchers are able to target the disease process at a cellular level, and it is through the help of naturally engineered defense mechanism. And if we look at this wonderful diagram, we can see that the Golgi apparatus is releasing the multivesicular body, which is being secreted out as exocytosis and the release of exosome. And on the other side, we can see the exosome uptake, endocytosis, where early endosome and then breakdown, and then the information is being relayed to the cell. Physiology of formation and degradation of exosomes is important for us to understand, that they release extracellular vesicles as part of their normal physiology and during acquired abnormalities. There are two categories, which is ectosomes and exosomes. And ectosomes are vesicles that pinch off the surface of the plasma membrane via outward budding and include microvesicles and microparticles and large vesicles. And exosomes, their size range is around 40 to 160, or an average of 109 meter in diameter, with endosomal origin. And depending on the cell of origin, EVs, including exosomes, can contain many constituents of cell, including DNA, RNA, lipids, metabolites, cytosolic, and cell surface proteins. Again, this is just to reiterate what we just spoke. We can see the sequential invagination of the plasma membrane, resulting in the formation of multivesicular bodies. It intersects with other intracellular vesicles and organelles, which contributes to the diversity in the constitution of exosomes. And currently, the physiological purpose of generating exosomes remains largely unknown and needs investigation, and it is still a speculative role. This is a very nice cartoon to show how the materials is collected from body fluids, cell cultures, supernatants, plasma, and through the identification based on physical and morphological characteristics, these are purified or these are isolated. And based on that, we do the testing and the analysis. And then once it has achieved that level of QC, the final exosome product is released for therapy. And this can be stored at minus 80 degrees centigrade or under cryopreservation or lyophilization, and then shipped to the hospital for the treatment. The challenging procedure is to procure exosomes of high yield and the fineness that it requires for a nanoscale vesicular component to hold that kind of information inside these exosomes. Now, separation can be made by several methods. Some of the most common is ultracentrifugation, ultrafiltration, gradient ultracentrifugation, precipitation. We see chromatography, mass spectrometric immunoassay, magnetic activated cell sorting, which is one of the newest technologies currently in the market, and microfluidics-based techniques. Again, this is a great cartoon to show how the different sources, such as umbilical cord, adipocyte, bone marrow, the cells are collected, the cells are expanded in the cell medium, the culture substrate is collected where the exosomes are found. The exosomes are isolated through one of the processes which I mentioned earlier, such as ultrafiltration, ultracentrifugation, mass spectrophotometry, or any of these. And then the exosomes are characterized based on the particle size and the concentration. Based on nanocyte transmission, electron microscopy, we look for total protein content and the protein marker. Sorry, again, as I mentioned, that various methods of isolation, confirmation, quantification, and analysis are present. And due to that, it has its own set of advantages and disadvantages. And it brings a lot of heterogeneity in these analyses. And this is the reason why exosomes need standardization. And some of the complex proteins in the exosomes are analyzed by the processes like Western blot, flow cytometry, mass spectrophotometry, high precision fluorescence. In the nanoparticle tracking method, we can look at, or we need to look for resistance pulse sensing method, microRNA processing, RT-PCR, and confirmation by electronic microscopy. So this is again a great cartoon to show which are the different protein exosome markers such as tetraspanins, CD9, CD63, CD81, then ALEX tag 101 in the exosome formation. So we can take a look at that. Now, what is important for us to understand is the different methods of uptake of exosomes by the target cells. So based on the type of exosome and the cell that it is targeting, it has different methods of being uptaken by these cells. One is the clathrin-mediated endocytosis, calveoline-mediated endocytosis, lipid raft-mediated endocytosis, phagocytosis, pinocytosis. It can lead to a membrane fusion uptake or receptor-mediated uptake. So these are the different uptake mechanisms or mechanism of action. And sorry for the repetition of the diagram, but I think this, in a nutshell, shows us how a cell-to-cell transit system works in our body and how these there is a balance of immune response and regulation of central and peripheral immunity. At the same time, metabolic reprogramming and regulation also happens. And then there's waste management or host microbiome interaction and viral immunity. So all of them play an important role as a hallmark of exosomes. Now, there's obviously an advantage of the the EV or exosomes over stem cells. The superiority of the exosomal cargoes compared to the stem cells on clinical and therapeutic potential is noted by lack of inherent risk associated with any cell-based therapies, including stem cells. There's a lack of replicating potential and the risk of malignant transformation is minimal. Lack of immunogenic response towards infection and cancers, targeted action at the site of interest is very important and crucial for this. Currently, we have data of around 79 clinical trials which have been registered among the global researchers in various pathologies, and 11 clinical trials have been completed where they have shown positive diagnostic and therapeutic effects of these exosome-based treatment. Sorry. Exosomes, due to their different mechanisms of uptake and these potentials, are being used as drug delivery tools, and their development of targeted drug delivery system is to harness the potency of the therapeutic products. So you can give a special attention due to their naive characteristics derived from the parent or the host cells, and ECVs play a major role in intervening or a disruption of the pathobiology of disease processes through the intercellular signaling cascade. It also offers neuroprotection and neuroplasticity since it shuttles around the blood-brain barrier, and this is why it is a good candidate for treatments or therapies for neurodegenerative diseases. The versatility of EVs enhances the intracellular signaling capabilities and their movement across cellular membranes to restore the micromolecular homeostasis. And currently, polymer-based nano-delivery systems are being developed. So based on all this background, there's a lot of potential therapeutic application of exosomes. And as I mentioned, nervous system, neurodegenerative disorders, spinal cord injury, aesthetics for aging, cosmetic use, lungs during COVID-19. We saw a lot of use of exosomes, heart, kidney, pancreas, gastrointestinal systems such as the irritable bowel syndrome, Crohn's, for joints. So we do see a lot of exosomes being used, or the clinical trials being run for rheumatological disorders, orthopedic disorders, and some of them for reproductive systems such as infertility. But the most common routes of delivery is IV, and despite its clearance in the liver and the kidney, it's widely used in orthopedic, neoplastic, and cardiovascular pathologies. Intramuscular subcutaneous are chosen due to the ease of the injectable area and the dosing volume, and it's mostly used for the neuromuscular or musculoskeletal condition, aesthetic, and cosmetic indication. However, we do see use of exosomes through intrathecal routes, especially in the neurodegenerative conditions such as Alzheimer's, Parkinson's. During COVID-19, localized aerosol sprays were very important for helping patients. Management of wounds and ulcers can be done for hair growth and rejuvenation in age-related therapies. Since it has all this potential, we really need to have a formulated guideline to foster their clinical usage. So International Society for Extracellular Vesicles, which is ICEV, was formed. European Network on Microvesicles and Exosomes in Health and Disease, which is MIHAD, and US FDA are currently the pioneers in providing us the guidance. There, however, remains an issue with the categorization of any emerging novel therapeutics for humans. Global regulatory requirements. A novel drug, when it is being developed, needs to strategize to standardize. And that is basically how we can validate the proposed technology in various labs that are developing this technology. With the help of these policies, the potential of the exosomes can be utilized at appropriate standards. And the SOPs are basically for collection, processing, testing, quality control, and the manufacturing of exosomes for clinical usage. There are currently no FDA approved exosome products for human use in the USA. The exosomes are currently classified as a 351 product for safety, efficacy, along with purity and potency. It's in the investigational new drug development phase, and the regulatory framework addresses the safety standards for the production and quality control, especially for microbial viral contamination, and it demands the GXP standards, which is good manufacturing, good lab, good distribution, good clinical, good scientific practice. Now, there is a pharmaceutical category of exosomes, and CBER, or the Centers for Biologics Evaluation and Research, they are regulating it as a biological product. So based on the individual type, the framework was laid down for products in this category applies to the exosome. So for example, an anti-tumor vaccine that uses exosomes will be regulated under the regulations provided for therapeutic cancer vaccines. So the functional moiety, or the functional part in the exosome-based therapy, determines its medicinal type. And hence ICEV has provided further classification or further categorization by therapeutics acquired from unmodified cells, therapeutics acquired from genetically manipulated cells without transgene, therapies acquired from exosomes and gene-modified cells with transgene classified as gene therapy products, or native exosomal therapies are used as drug delivery systems, as carriers for the biological and chemical components, and are considered as biological medicine. Sorry. In the UK or in the Europe, they have been classified as ATMP, or Advanced Therapy Medicinal Product. Again, it's subgrouped according to the conventional biological medicinal products, such as biological properties, physicochemical and immunochemical properties, and it includes the somatic cell therapy, gene therapies, and the tissue engineered products altogether. ATMP therapeutics involve products that have been more than minimally manipulated. It's non-homologous. An example is the hematopoietic cells for orthopedics, which normally would not be consistent with the regulatory approval. Nucleated and viable cells are present in the product, and products with therapeutically active transgene from genetically engineered cells are considered as ATMPs. Now, EV-based therapeutics are produced from human material by a manufacturing process comparable to the ATMP production, and this is why we require a GXP or a GMP lab to process these. Now, based on all of this background, ICEV has changed some standards, and they have put in a minimal requirement for exosomes or EVs, isolation from extracellular body fluids or conditioned cell culture medium, quantification of at least one protein from 3 different categories, characterization of single vesicles within the EV preparation using at least 2 different technologies, electron microscopy or atomic force microscopy for imaging and nanoparticle tracking analysis. So they have now started to further classify us, and based on that, EVs are further classified as exosomes or microvesicles based on their intracellular biogenesis pathway. And as we saw the microvesicles earlier, we now know that exosomes are formed within the multivesicular bodies at the endolysosomal pathway and secreted upon fusion of MVBs with the plasma membrane. So let's look at some of the companies that are there. So despite this flurry of activity, exosomes are coming under increasing scrutiny with the public safety notification on exosome products, and caution is required. And currently there are no FDA approved exosome products. So Eagle Therapeutics is one of the first in this class, phase 1/2A ready. It is from allogenic bone marrow derived mesenchymal stem cells. It treats burns and epidermolysis bullosa. This was the first IND application to begin clinical trials in burn patients. And then we have Athlon Medical, which is a hemopurifier, which can capture tumor-derived exosomes underlying several forms of cancer. Then Angarium Biosciences has a hybridosome platform, which is helpful to employ the potential of exosomes in RNA-based applications, such as RNAi, mRNA replacement, and gene editing. Sorry, Aruda Bio is again a pioneer, and it's harnessing the natural abilities of neural exosomes to cross the blood-brain barrier and enhance the body's anti-inflammatory, self-repair, and protective mechanisms. That's why being explored in treating neurodegenerative disorders, and it can cross the blood-brain barrier and enable drugs and drug combinations to approach the target cells and treat the patient's neurological disorders. Capricorn Therapeutics again is undergoing a clinical trial, CAP203, comprised of exosomes isolated from the company's proprietary cardiosphere-derived cells. So Cellular is another company, Clara Biotech, which has its own lab tool, and in future we could actually be able to isolate exosomes in the labs at rates up to 100 samples per hour. Then we have Zodiac or Kodiak Biosciences, which is another platform which is very pioneering, Creative Medical Technology Holdings, Direct Biologics, EV Therapeutics, Eversome, Evox Therapeutics, Exco which is being represented out of Seoul, South Korea, is actually looking for innovative therapeutics for cosmetic products. So they're looking to regenerate or activate or deactivate various tissues or cells. And Exopharm Australia, which has its own Exosomes team, and they have a partnership with Rooster Bio, another company, which is again at the forefront of exosomes. So Exosome Diagnostics, Exosome Plus, Exosome Sciences, again, it's working on diagnosing and monitoring Alzheimer's disease, chronic traumatic encephalopathy. Sorry, Exosomics is looking at screening and molecular diagnostic test. Iliax Biologics, Invitrix, Chimera. Chimera is again having its own clinical trials for its SOCLO-TM product, which is isolated xeno-free, chemically defined, cell-free placental MSCs, and they are being used for cosmetic uses as well as orthopedic uses, skin repair, beautification, and regeneration. Lonza is again at the forefront, and it has many partnerships with different companies. And this is again another company that we would be looking for. Organicell, it's contains approximately 400 billion exosomes. Again, a nanocyte technology, region suppliers, Reneuron. Reneuron is a UK-based company. It has a CTX neural stem cell line under different culture conditions, which can be harvested at a commercially relevant scale. And Rooster Bio Inc., as I mentioned earlier. So we have stem Unicyte, Vivasome, SoStem. So there are many companies from different parts of the world, such as Australia, Switzerland, who are all working towards it, either diagnostic or therapeutic. This is a good table showing all of these companies targeting on exosomal research and their potential products for commercial use. And most of these are already under clinical trials now. So what are the challenges? The mechanism of action is essential for the clinical translation of therapeutics. And for us to understand what it is, we need to translate and identify what are the active substances, what are the properties, and the essential quality controls in manufacturing a clinical grade product. And although uncertainties exist on the MOA or mechanism of action, we need to look at the target cells. And currently, exosomes have not been accounted as high risk in an IND. So again, commercializing a large-scale manufacturing of exosomes-based therapeutics requires a robust quality management system, and we need a technologically superior facility, updated technology complying with GXP, GMP, GLP. And the endpoint of these investigational studies or INDs is to provide safety for the donor, the patient, determine efficacy, look at the potency. And unfortunately, at this time, we do not have a standardized protocol for isolation and storage. And many of them are homemade cocktails as protocols with no standardization for reagents, storage containers, and storage time for each desired exosome-based product. So in conclusion, isolation and characterization of exosome is a sophisticated technology. Ultracentrifugation is based methods are currently most commonly used. We do have limited commercial available exosome isolation kits, so isolation is difficult. For characterization, we do need very sensitive and very specific tests, such as the nanoparticle tracking analysis, transmission electronic microscopy, Western blotting, um, and we look at the type of the vesicle or type of the exosomes, and for the analysis of the size distribution. So we look at size distribution, morphology, and presence of exosome-associated markers. And despite some variability, we do see isolated MSC exosomes. They generally appear as a bilipid membrane structure, and 30 to 150 nanometer is the size distribution. And some of the markers that we're looking for are endosomal proteins, such as TSG101, ALEX, tetraspanins are most commonly, sorry, tetraspanins are more common family of proteins, which is CD63, CD81, CD9. And proteins in this family have 4 membrane spanning regions, and they are very specific to the plasma membrane. Again, it is, however, important to note that the exosome preparations are largely heterogeneous with variable level of undetermined purity that can influence the outcomes. And the positive side was the storage of the isolated exosome at minus 80 degrees centigrade until use did not appear to affect the function of the isolated MSC exosomes in those studies. So this observation does hold promise for a cell-free, off-the-shelf therapeutic strategy. So allogenic exosomes can be used for a much better utilization. And exosome surface markers and molecular cargoes have been worthily challenged. So it's already present. A lot of diagnostic biomarkers are currently being used. And due to their low immunogenicity, long half-life in circulation, and ability to cross the blood-brain barrier, exosomes have been explored as a nano-delivery system of therapeutic signals. And this is where the future research would really rely on. These are some of the references that I have used. Thank you very much for giving me this opportunity to present this wonderful topic today. I would like to acknowledge and thank the AATM leadership, the Cell Therapy Committee, Dr. Claudia Dufour, my association and my associates at AABP, AABRM, the Parents Guide to Cord Blood, IFATS, and the companies that I represent, Questa, InfoHealth, MEMP, and Regenerative Outcomes. And I always believe in us before me, ethics and social capital for global well-being. So we want to bring safe exosome therapies or therapeutic potentials to our patients and to deliver them safely. This is the social media that you can reach me on, and I'll be very happy to answer any questions. Thank you very much.
Thank you so much for your wonderful presentation, dear colleague. Here from Bangladesh, Dr. Kalyan Roy have some questions for you. The first one is, what are the adverse effects or side effects of this exosome therapy?
As I mentioned in one of the slides, the differences between the EVs and the stem cells, there are side effects. So we cannot say that there are no side effects because these are nanovesicles, but they carry the messages or the messenger RNA and DNA material. So there is a risk of cancers, but it is very less than what we would deal with in the stem cells. Challenge at this point is we don't really know because these are normally present in our body as well. So we don't know what kind of an actual effect that would have. And this is the reason why the US FDA or ICEV has started to study it more, saying if you're using them, especially in therapies or therapeutics, we need to study them more. So currently this is similar to stem cells, I would say, but a little bit less risk or less adverse events than what we face during the stem cell transplantation or infusion. But um, it's largely unknown at this time, if I can put it that way.
So the second question Dr. Kalyan wanted to know was, how much will it cost if approved by FDA, due to us being from a low middle income country?
Actually, sorry, you're talking about the analysis part, right?
Yes.
Can you? Yeah. So it depends on what kind of assays you want to put. So as Dr. Roy, we've already had some interaction earlier as well. So I can always say that if you already have a flow cytometer, then that can be done as well. So according to ICEV, they have not mentioned which assay you need to do. So based on your exosomes that you're producing, you can decide what assays you would like to do. So even if it's a low to medium, economic country, and not even all the Western, companies can do that as well. So many of them we just use as ELISA kits. There are some proteins which are available, so we can check against that. We'll mostly look at the counts that is available, and these can be done on a flow cytometry. So flow cytometry, I feel, is still more cost effective. It's not that expensive as it was maybe 5 or 10 years when we started using it earlier. But I think currently this is another challenge for us to bring in more kits, more tests that we can provide at a lower cost. And I think as the field will start increasing, the cost will also start reducing. But it is much lesser than what we would require for, let's say, a CAR-T therapy or a NK cell therapy. It's much lesser than that. And that is the positive side of all this, exosome-based therapy, especially the allogenic one.
Thank you, Dr. Asawari, for such a wonderful presentation. And if there are no further questions, this brings us to the end of the webinar. I thank our chair, Dr. Claudia Dufour, and I would thank again Dr. Asawari Bapat for joining us from Dubai. Thank you all. Thank you all the participants who made this, who took time out of their schedules to join us for this cellular therapy webinar. Ciao.
Absolutely. Thank you so much. Thank you for having me here. Bye-bye.
I think,
reach out to me if you have any questions. More of the series in future.
Last question, Asa. Many cord blood banks have cryopreserved mesenchymal stem cells from cord tissue. Do you think it's possible to obtain exosomes in the future in this kind of mesenchymal stem cells cryopreserved?
Absolutely. This is why I mentioned. So few of the companies that we are already consulting are already doing this, and they're doing it in clinical trials. So there's not just a hypothesis, but they have moved into clinical trials, and these are being done from the cryopreserved cord tissue or cord blood, which was probably stored 5 years, 10 years, or 20 years back. So this is what it has shown, that even after processing or even after cryopreservation, we can still extract the exosomes out of these sources and use it for therapies. And this again would be a bigger potential of exosomes, because then we can utilize all the samples or all the products that we have saved a long time ago, and we didn't know how the cord blood or the cord tissue is going to be used. So this has shown that we can thaw the sample, process it, and then use the exosomes from these sources as well. Did I answer your question?
Yeah, and do you think it's probably differences between this naive exosome from this tissue, placental or cord tissue, if you compare with bone marrow but an adult?
Oh, yes, that's a very good question, Claudia, and thank you for asking this, because as I mentioned in one of the first slides, that the exosomes, or the EVs as we call it as, their content depends on the host or the source of the cells. So based on the source, it is always better to choose what kind of a condition you want to treat, what kind of a therapy you want to act, because based on that, you want to choose the source of your exosomes. So let's say, for example, I can give you a very good example, someone doing a placental or a cord tissue or a cord blood, exosomes will act very well on all the hematological or even orthopedic uses, because that's where you need the neovascularization and, the development of bone and other tissues that we're looking for. So the regeneration is much more faster when we look at the placental exosomes or the cord blood or the cord tissue exosomes. So there's definitely a change in that from different kinds of exosomes and, the potential how we can use that.
Okay, I I saw that is an incredible option right now that in the future. And I have the last question. And right now, at the moment, to reduce the stem cells for cryopreservation at the placental blood or the cord blood, we have wasted the plasma at the moment to reduce for cryopreservation. Do you think it's possible to obtain for this plasma discarded exosomes or other bioactive components?
That's a great question. And if I can put it this way, there are companies which are currently exploring exactly that. So maybe in future, even the plasma that is discarded out of the cord blood will not be discarded and will be considered as something that we should, explore, and something which will be stored in future as well. Because yes, there is a potential, there's a huge potential, because what we are really finding out, and I think again, it ties down to Dr. Islam's question earlier, that because we are functioning in a not known territory, we need to study this more. We need to know this more about how they actually react. So one is, we have realized that there are exosomes in these sources, but now we need to study on how they will react in the body once they are, inside or in vivo. And there's a lot of study which is going on now for the cord blood or especially from the naive sources, because we want to know if the plasma or the bioactive substance which is already present, can it really help you in providing. So I have come across a paper, and I can share it with you when I can look into it properly. But I think it talks about how naive cord blood has helped a lot of people in China, especially during COVID. And when you look at the cord blood, if it is just a blood transfusion, then what was different than a blood transfusion or a cord blood that was given? And then the hypothesis is maybe because of all these bioactive substances which are present only during the birth of the baby, or these being passed on from a mother to a baby, that is what is the special, liquid active substance which can, help you in providing these therapies. So yes, a lot of potential, a lot of thoughts. And yes, I'm open to a lot of projects, a lot of, activity going on in this area currently. So this will be something which I can explore very well. Thank you so much, Claudia.
Okay, thank you. Well, that was a wonderful activity. I would like to offer our thanks for the AATM Cellular Therapy Working Group, also for all the audience, and of course to my colleague and friend, Dr. Aika Hindal, who works very, very hard in this activity. And of course, Asawari, once again, very nice to meet you, and thank you so much.
Thank you.